Hello, everyone. Hello. I think we're going to start now. Thank you all for coming. Thank you very much. It's really exciting to be here. I'm Pablo. I'm the president of the Systematics Association, and this is our first in-person event in 3 years. So this is super good. And I see many new faces, which is also great. If you don't know what the Systematics Association is, we are 80-something years old and a scientific society interested and supporting all aspects of research and taxonomy and systematics in the light of evolution. We have conferences, we have publications, we provide grants. We're going to start offering workshops and other initiatives. So, and our membership rates are super affordable. Have a look at our website. You will see it is very easy to find. If you type Systematics Association on Google, have a look at our website, see all the things we provide. For just a bit of promotion before we start the meeting today. In a month's time, more or less, we have the Young Systematics Forum. It is an online event. It's free to attend. And it's a one-day conference on systematics aimed at very young systematists. So people in the PhDs or the master's thesis present there often for the first time. So it's often it's a really nice place. If you want to attend, also look for it. Abstract submission closes today, so you won't have a time to submit it, but you can still register to attend the meeting. And that's all the promotion for now. Thank you again very much for coming after the lecture now. We will go upstairs to the library for a wine reception. And our speaker today is the Founders Lecture, Systematics Association Founders Lecture. We've invited Rafael Medina. He's an assistant professor at the Complutense University in Madrid. He did, I know this pretty well, he did his PhD also in Madrid, but in a different university, in the Autonomous University. And then he finished in 2012, I think. Moved to the US, to the University of Connecticut, to do a postdoc there, all of it working on bryophytes, different genera or different families, all of it on bryophytes until 2016, when he moved to Augustana College in Illinois as an assistant professor. He worked there until 2020, and during his time there, I don't know if he'll be talking about that or not today, but during his time there, he also became, he was an assistant professor, but he also became curator of the herbarium, and the herbarium there became very active during those years. So that's also something very exciting to read about. In 2020, Rafael got a position back in Spain in a different university from his PhD as an assistant professor, and he's been working there since then. So he started in the middle of the pandemic. And also very recently, he's moving, not only studying bryophytes, but he's also started studying ferns. So he's quite an interesting research object of research. Apart from that, Rafael is the president of the Spanish Biological Society since last year, and a really engaging speaker, I've heard. It's me. So all yours. Thank you very much for being here.
Thank you again for that presentation, and I'm very grateful to the Systematics Association for giving me this opportunity for sharing with you some of the stories with the high achiever low plants or bryophytes. Can you hear me okay? Okay. So I'm honored of being here in this celebration of science. So let's begin. This is me when I was 20 years old. And I don't know if you can tell from my outfit, but I was absolutely obsessed with the natural world. Okay, it was, I didn't care if it were butterflies or birds or flowering plants or ferns, everything was exciting. I was just, you know, being like that for a number of years. I bet that many people here in this room feel related to that, right? At that time, I was an undergraduate student at the Universidad Autónoma. I was trying to get my degree in biology, and I really wanted to make a career out of this, right? And at that point, it's like you feel you have to choose. And again, this was a hard decision because I was interested in all the natural world. I was convinced that any sort of organism would be fascinating by itself, plants or animals. But there was only one caveat. If I became a botanist, I would study actual plants and not those bryophytes that were so frustrating because it was impossible to identify. Okay, and I'm here now going to, I have been making a living in the past 15 years or so, studying and staring at moss and bryophytes and very small plants, sometimes smashing them and getting their DNA, that type of thing. But this is actually something that is very common even among people that study bryophytes. It's rarely a love at the first sight thing, because they are so underappreciated and it's so difficult to get to know them that I'm just concerned that coming here and talk about bryophytes is going to give you, impression of something very obscure. And in fact, many bryologists have, communicated this type of worries, about our subject organisms with time. So with this presentation, I have a twofold objective. Number one, I would like you to know bryophytes a little bit better and perhaps get rid of some of the biases that you may have about tiny plants and their interest. But I also think that even if you are not very into bryophytes or botany for that matter, they have something to teach you. They have something that we can all learn about in terms of what's the biological importance of complexity and simplicity. So in my the first part of the presentation, I'm going to make an introduction on bryophytes in general and show you that in the recent years, there is a lot going on in terms of exciting research that is changing the way we see bryophytes. And then the second half is going to be more about the family, the particular family that has been, giving me some headaches during the past years. So if I see bryophytes, many of you will immediately come up with, these sentences that you read in botany textbooks, right? Like non-vascular plants, early land plants, and my favorite, lower plants. Okay? All these qualifiers are to some extent incorrect or misleading. So we will go one by one. First, what are bryophytes? So when I say bryophyte here, I'm using a very broad umbrella word that will refer to three monophyletic group of plants. These are land plants, these are embryophytes, mosses, liverworts, and hornworts. Probably you have heard of mosses, there are two more. They are very different, but they share some traits that they have in common. First, well, they are small plants, okay? They are very simple structurally. They are not going to be as complex as the flowering plants that perhaps you have in mind. And you may remember that all plants have two generations, right? They are going to alternate a sporophyte. This is the diploid generation, 2n number of chromosomes, and a gametophyte, the n generation, n number of chromosomes. Bryophytes are special because the gametophyte is the one that is the dominant generation, the one that will be doing photosynthesis, so on and so forth. The sporophyte is very simple. Most times it will just be like this, a stalk or seta, and then a capsule or sporangium where the spores will be produced. These plants release spores, they don't release seeds. They only have one sporangium per spore. That's another thing that is unique for these plants. And you have heard that they are non-vascular plants. What does this mean? Okay, it means that they are not supposed to have a vascular system, same way that you have arteries and veins in your body. If you want something to become big, even if it's a plant, you need water conducting cells. Bryophytes sometimes do have vascular tissues, and this is one misconception that we need to get rid of. Sometimes you will see bryophytes like this. Actually, most of them will not have vascular tissues at all. It's partially true. If you make a cross-section of a stem, there is no specialization in any sort of water conducting cell. But a lot of mosses do have water conducting cells and tissues. The point here is that they don't need it to survive. If for whatever reason, these structures here become empty, they lose the water, you have bubbles there, whatever, the plant will not die. That's the difference. It will not, if they have it, their life is not dependent on it. Now, for comparison, let's think of a humble flowering plant like this Arabidopsis, they will also have an alternation of generations on sporophyte and gametophyte, but the sporophyte will be the dominant generation. It will have a vascular system, vascular tissues, that are critical for their survival. If for whatever reason air bubbles appear in enough quantity in some of these vessels, the plant will die. Technically, we'll have several sporangia rather than just one. These are just details, but the point is, when you see a bryophyte and you see a vascular plant, there is a difference in terms of the structural complexity. That's why you can have a giant sequoia, but you are never going to have a giant moss in that for that matter. The problem here is if we make the assumption that something that is structurally simple is also evolutionary unsophisticated. And I have to apologize in advance if you think that you will never make such conclusion, or you would reach to that conclusion by yourself, but you will be surprised of how often I find among my colleagues this type of thinking. This is not true, but of course you may think that bryophytes, just because they are so simple, they are primitive, they are very old, and they have remained unchanged for long periods of time. We have a lot of examples following this narrative. You may assume that bryophytes are relics from the past, that they don't evolve fast anymore, they are no longer in their prime. They had their era, now it's the era of the flowering plants. They are just relics, right? They will have slow evolution, for example. Fast evolution is something that you expect for flowering plants. If you are at all into evolutionary biology of plants, you will know that polyploidy is a huge topic. It's a very hot topic right now. We know that it's a source of genetic innovation and evolutionary innovation for all sorts of vascular plants, for example. In this review paper from 2015, it was assumed that bryophytes would have very low impact in terms of polyploidy. And we will see that this was made without any support. This is just following the narrative that just because something is simple is not going to be sophisticated. Let's dig in this concept. And in the past years, and I'm just talking about the past 4 or 5 years, we have completely changed the way that we see bryophytes. But even before that, to me, the best way to explain why this narrative should be changed is going back to the plant tree of life. The land plant tree of life used to look like this for the past 20 years. This has been the standard during from the beginning of molecular systematics. This is the tree that I learned in school, probably the one that you are familiar with too. And you may think that the topology of a tree is not going to convey any subjective story, it's just going to convey what happened. But actually, some topologies convey some narratives in a better way than others. And in this case, there is a narrative that is very clear. We have 2 leaps in sophistication. The first one is the origin of land plants themselves, right here. And then we have the next relevant evolutionary step, which is the vascularization, the origin of vascular plants, okay, right here. All bryophytes are resolved in a grade, and this means that the different common ancestors, the different intermediate nodes of this tree are going to be bryophyte or very bryophyte-like. It just makes sense, right? And then, of course, this also means that mosses are non-vascular plants because they missed the synapomorphies that made vascular plants possible. These nodes were missing these synapomorphies over there. They are lower plants because implicitly this tree is giving you the impression that there is like a staircase to progress, so to speak, and the bryophytes are the intermediate steps, the one that became, just in that position. And you can also say that they are early land plants because the very first land plant was either a bryophyte or something very similar to a bryophyte, right? This is everything that this tree is conveying. And in fact, I just said that this goes back from the very beginning of the molecular systematics, but this is even older, more than 150 years. Ernst Haeckel, who became, probably most of us remember him because of the issue with the fabrication of the embryos, but I think he deserves a lot of credit because he was, as far as I know, the first person to try to reconstruct a first universal tree of life, just labeling phylogenetic trees and writing names of those branches. And his narrative was exactly the same that I was mentioning earlier. regarding bryophytes. They are the intermediate steps between the algae and the first vascular plants. In this wonderful phylogenetic tree of plants, this is exactly what you see, a bryophyte. Okay? Now, how is this changing? In the past years, very recently, we are having more and more evidence coming from different labs and different groups all over the world that suggest that bryophytes are aclate, that they are monophyletic. The source of this information is either because we are able to analyze more amount of data, but also because we are getting rid of some biases that were impacting our previous trees. Again, this is not just one group of one research group. These are several people saying the same thing. Now, let's see how this difference impacts now the story. We have our origin of to all land plants. And then we have the common ancestor of all plants right there, which is not necessarily a bryophyte, because now the ancestor of all bryophytes is in that position that you can see there. This opens the door to a very thought-provoking perspective in which the origin of vascularization is not necessarily unique for tracheophytes. And chances are that bryophytes are simple, not because they missed those synapomorphies, but because they lost them secondary. Now the affinity, the placement of vascularization is not clear anymore. And we have other sources of scientific information that is supporting this perspective. Again, several labs, several groups are showing that the genetic signature of some of the advanced vascular functions that you expect to find only in vascular plants was already present in bryophytes and sometimes also in streptophyte algae. So they predate the origin of land plants themselves. I like in particular this paper here that demonstrated that in one of those mosses that supposedly have the fake vascular tissues that I mentioned earlier, they can withhold negative pressure that is comparable to that you can find in vascular plants. They don't use it the same way, it's not essential, but this gives the impression that this is an atavism that some mosses kept for whatever reason, but their ancestors were perhaps vascular or protovascular, or at least not merely bryophytic. The most recent study that is also supporting this point of view came just 15 days ago. We published this in Nature, Ecology and Evolution. It also supports the monophyly of bryophytes, but it also reflects that the enrichment of new genes that vascular plants have appear right here in the origin of all land plants. And what happened in the stem, in this branch that leads to bryophytes, is that there was a significant and very remarkable loss in some of these gene functions. So it's like natural selection was selecting against the conservation of these complex and elaborated traits in the case of bryophytes. They were doing better without that. And of course, the question is why? Well, chances are that bryophytes got adapted to a particular niche in which they didn't need all this structural complexity. It was completely useful. You all know about tardigrades, probably, right? We all love tardigrades. They are almost immortal. We know the story, but tardigrades are not that special. Tardigrades are just one more members of a whole type of ecosystem in which life and death doesn't have a very clear frontier. You have that in animals, you have that, rotifers, that type of thing, right? Nematodes, but also creatures such as lichens, and of course, bryophytes, who can also remain in cryptobiosis for long periods of time and then coming back to life. I'm sorry, this should be an animation. Apologies for that. This is called poikilohydri, and this is actually what mesmerized me about bryophytes and what made me to fall in love with them. So it's something I try my students to see with their own eyes. When you put water for the first time in a bryophyte that has been virtually dead for, who knows, years perhaps, right? And coming back to life. So definitely this is an interesting trait that natural selection will want to keep, no matter what happens with that. And we have some spectacular examples of bryophytes coming back to life after 400 years under the ice. And in a more close environment here in England, we have demonstrated that the spore bank of Fiscomitrium euryostomum was able to survive more than 100 years in the sediments. Okay, so what does this narrative say? To me, after the origin of land plants, we still don't know how the first land plant was like. There is definitely a trend towards vascularization, towards gaining a high maintenance vascular system that allows you to grow in size and to do things that are very spectacular and that we admire in plants. But there is also this alternative way of being a plant in which the size of the vascular system is not that important, and you focus on being low maintenance and your capability of being polyploid. Equally interesting, but maybe not so spectacular from the structural point of view. So let's review these terms. Should we call these lower plants? I mean, it's fine if we call them low plants. There's nothing wrong with that, right? Their size is limited. It's like you almost have to choose. You can't have both branches at the same time. You have to opt out some of the abilities if you do one thing or if you do the other. Try a giant sequoia to be polykiloidric and let's see what happens. Early land plants. Early land plants, it's more and more clear that it's not correct because the very first land plant perhaps was completely different. It was nothing like a bryophyte. So we should stop thinking that the very first land plants were bryophytes or bryophytes-like. We don't know. We need fossil evidence. It's not coming yet. And then vascular plants, I mean, it's okay, but we have to keep in mind that perhaps they lost secondarily some sort of vascular system. And it's true that most bryophytes will not have a vascular system, but just with that caveat, it's not something that they never had. Okay, I hope that this grew some interest in bryophytes and helped you to see it in a slightly different way. Let's dive in now the family that has given me some headaches for the past year. That family is the Funariaceae. Funariaceae are interesting because they are going to challenge a general trend in moss systematics. You have there an arrangement of different moss species, different sporophytes, and if you pay attention, you will see that there is a huge variation in terms of how the capsule, the single sporangium of the sporophyte, opens and releases the spores. You have, for example, capsules that have like a spiral line of the dehiscence. Some of them will just pop their operculum that will just be thrown away. You have capsules with several lines that open. There is a variation in here. This is, in fact, one of the most conserved traits in moss evolution, the structure of the sporangium, the structure of the sporophyte. It's what defines the backbone, and it's like the trait that you will focus if you want to identify a moss that you don't know what it is. If you go out there to the field and you see a plant, an unknown plant, you find the flower, you dissect the flower, you count the petals, you dissect the carpels, etc. And if you are an experienced botanist, it will let you know which family it belongs to. If you are a zoologist, you work with vertebrates and you see a skull, probably you can just look at the structure of the bones and you will say, okay, this belongs to this group or this group. Okay, you do the same thing with mosses, looking at the structure of the spore. And in particular, there is this evolutionary trait that most of the mosses will have, a hygroscopic peristome that can open and close. All these are critical. Okay, the family that we have been studying for the past years, it's very different. You have about 300 species. It's present all over the world. And the variation that you can see in the complexity of the sporophyte is very surprising. Okay? I'm going to give you a tour and to present some of the Funariaceae, some of these bodies, and trying to explain how their sporophyte works. And you have to remember that this is the old genetic circumscription. So until more or less recently, there were these genera. There were 3 genera that had a good number of species, like 80 or 90. These are Funaria, Entostodon, and Fiscomitrium. And then something weird. There were 13 genera. I'm going to call them the minor genera with up to 20 species. So most of them are only going to have 1 species. Okay? Let's go one by one. This is Funaria, perhaps one of the most complex sporophytes in all mosses. To begin with, the zeta will be hygroscopic. So depending on the humidity, the atmospheric humidity, it will curl up and down protecting the capsule if it needs to, or allowing a more efficient spore dispersion. The capsule is sealed by a ring of cells that we call the annulus, the revolving annulus, that is quite unique, because once the spores are mature, and only when the spores are mature, the very first time that this annulus touches liquid water, it will open. So the cells will expand automatically, and the operculum or lid will fall, just like that. Once this happens, you will see the peristome, a hygroscopic peristome that in this particular genus is also very unique because it has this shape. It's like a diaphragm of a camera. And this structure is also hygroscopic. This is not a time lapse, this is real time. So when the conditions are right, it will slightly open so that you can release steadily, but with some control, the spores that you have. This is as complex as you can get for a bryophite. This is one of the most sophisticated methods of spore release. Okay, you can think of this as a cake box. So you have something inside, you have some content, and then it's more or less like wrap that all nicely, and you can open and close it depending on what you want. Okay. Now I'm jumping to genus number 3, Fiscometrium. Also like 80 or 90 species, and the mechanism here is very simple. You just have an operculum, and the operculum falls. There is no annulus, there is no peristome, nothing. It's like a sugar bowl. You open and there is no control. All the spores will fall. This is Fiscometrium. I'm not going to talk too much about particular species, but I want you to keep in mind the name of this one, Fiscometrium piriforme. This is perhaps the most boring moss that you can find. It's a no-brainer. You identify it when you see it. It's present in roadsides in North America and Europe. Nothing exciting about it. Okay, I want you to keep this in mind. This is a little bit like the shepherd's purse or Capsella bursa pastorum. Do you know this? A very common species on the roadsides. It's like you will never look twice. It's like it's there. Okay, I don't care. That's it. Okay. Major genus number 3, Entostogon. This is a little bit tricky because there is a huge variation. So you have some some species that are almost like a mini Funaria, so cake box, and you have others that are like a sugar bowl, and then you have a range of intermediate forms with some sort of peristome. Some of them are functional, some of them are vestigial. Okay, so for the sake of simplicity, I'm using this paper box structure. There is some type of control on how the spores are released, but that's right. Okay, these are the 3 major genera. Let's go now to the minor genera. Remember, up to 13. For example, Funariana. This is basically a Fiscometrium, a sugar bowl. The operculum at some points falls, and that's it. There's nothing else to be done. Now, pay attention here. The zeta is very short. This will accelerate the production of the spores because you just don't need to grow so tall, right? You just produce the spores and that's it. A Phanorrhegma. In this case, you can see that the capsule is like a sphere. It has no operculum. You need to break it to release the spores. But it will have a line of minimum resistance where the cell walls are very, very thin, right here. So you break it, but at a designated place. Think of this as an ampoule with some medicine or something. You have to break it, but always the same place. Last, but not least, just a few examples. Piscomitrella, up to 30 different species were described under this name. The capsule is a sphere, and you need to break it too. And this time there is no line of dehiscence whatsoever. It's like a neck. You just need to break it wherever, right? The spores will not be released until the capsule is broken. Now, this name rings a bell. There is no coincidence because the species formerly known as Fiscomitrella patens is the flagship system organism for the study of bryophytes. The first genome that was sequenced belonged to this species, and well, it has a lot of advantages for that. Remember, I just gave you 3 examples, but there are up to 13 genera, okay? 13 genera that are 1 or 2 species with a very reduced peristome. And of course, the question is, how is this even possible? I just told you that the structure of the sporophyte is one of the most conserved traits along moss evolution. There is something going on here. If this was a flowering plant with this shape shifting in the flower structure, or a vertebrate with, you know, this very variable position of the bones, we would all be like completely obsessed of what's the story behind it. So this is one of the questions that we wanted to address. And the other one, of course, is, does this generic treatment make sense? because perhaps you can just merge together all the minor genera, maybe this is better, or perhaps the right thing from a phylogenetic point of view is completely different. We didn't know at this point, but something should be done. And this is what we did, right? So of course there were a number of people involved in this project. I want to mention especially those who I have worked more closely with and that have been responsible of the latest developments of our work with the Funariaceae, beginning with Bernardo Finea at the University of Connecticut, who was my mentor in all this Funariaceous stuff. Matt Johnson from Texas Tech University, he's the bioinformatic guru that made possible everything that you're going to see later, and Nikisha Patel, who has taken the lead of the most recent study on the Funariaceae. Our approach, after some previous, I wouldn't call them failures, but Sanger sequencing was definitely not enough for this family, was using this technique that is called HiveSeq or target capture. Are you familiar with it? You are, okay. So this is great because it takes advantage of high throughput sequencing with Illumina technology. Basically, you're getting loads and loads of sequences, but you are not going to sequence random parts of the genome. You can choose which parts you want to sequence. And we were using two sets of baits because what you do is basically use some oligonucleotides that will fish from your genomic library only the regions that you want to. We use two of these sets. The first one, all the coding genes of the mitochondrial and the plastid gene, and a second one with a selection of 650 nuclear genes. Okay. Our first iteration was to use the organellar genomes, so only the 123 genes from the mitochondrial and the plastid. You may think that this tree is unresolved, but it was actually resolved. The problem is that most of the richness of this family came after a rapid radiation that happened about 28 million years ago, relatively recent. The notes here are so short because there was almost no information in these different cryogenic events. So everything happened very, very fast. Second conclusion that we reached here, the classical genetic circumscription needs to be rethought. Perhaps with the only exception of Funariana, the cake box thing, that stayed clearly apart from everything else, it was clear that that was a monophyletic. But then we have this grade of Entostodon. Piscometrium looked more or less monophyletic, but then we have this Entostodon fungaricus in the middle. And then, of course, all these reduced forms that you can see here, of Apanoregma, Piscometrium, Funariana, etc., they will scatter throughout the phylum. So they are not a good predictor of systematic position. Remember that super boring most species Piscometrium piriforme? Okay, so this is also scattered here as a polyphyletic acid. This iteration of this study maybe left us with more questions than answers, so we were really eager to try our second data set, or our second bait set with 650 genes. A few notes of caution before working with nuclear genes. Well, first, this was possible thanks to the bioinformatic pipeline called HivePiper, developed by Matt Johnson, that has a number of advantages. First, from the reads that you get, of course, you will be able to retrieve the original sequence, the original locus that you wanted to sequence, like this one. But HivePiper also allows you to fish some sequences from the splash zone, so the areas that belongs either to the introns or to the flanking regions. And this was very, very valuable because those regions are less conserved, and they gave us more information that was particularly useful in a rapid radiation. When we had to analyze this data set, it was relatively trivial to get a tree that was technically optimal under different conditions. But you also need to take into account the conflict between the different stories of the individual genes. So the individual gene trees might be in conflict with the most likely tree at the very end. The way you are going to see this visualized is each node is going to have a number of pie charts. So when you have a blue area that is very large, that will say that most gene trees agree with that node in particular. Thumbs up, everything is fine. But if you have a lot of pink area, that will mean that despite that node being the most likely, a number of individual gene trees do not agree. Okay, so that's the legend for that. And finally, Piper will also detect parallelism. So if you have a locus and there are several sequences that are not identical, it will let you. Why is this important? Well, because this is one of the evidence that, very strong evidence that you might have hybrids or polyploids, allopolyploids. So we have to take care of this in a later study. This tree here is only showing the Funariaceae without Funaria, that was already sorted out, right? And without the hybrids. First conclusion, using introns is very useful. It's great. It will increase the resolution of the different nodes. You see the two pie charts in each node. The first one is without the introns, the second one is with the introns, and you will see that the blue area gets larger every time. So that was useful. We also see one clade, possibly a future genus that gets segregated from Entostodon. This is more or less easy, very clear. So we are working towards a new generic circumscription. And we did the same thing with Fiscomitrium. So Fiscomitrium, it's a monophyletic group right there. But at this point, it was very clear that some of these extremely reduced sporophytes were not something systematically useful in terms of the genetic description. This was just a syndrome. So independently, a lot of these different species look very, very similar for some reason, but they don't deserve to be in a separate genus if we want to be consistent with the systematics. That's what we call it the Fiscomitrella syndrome. Fiscomitrella, Aphanorhegma, Fiscomitridium, and a few others. This was the graphic abstract that we wanted to submit as for this paper. I'm mentioning this here because there was some level of I don't know, frustration, because a lot of people were working with Fiscomitrella as their model organism, and maybe if systematics is not your cup of tea, you don't understand very well the changes in nomenclature and that type of thing. I think we are fine now, and that people are accepting Fiscomitrium patents as the name for this species. You can still name it Fiscomitrella as a nickname if you want. But to me, it's important that we give it this name because we are conveying some information that you would not have if you call it Fiscomitrella. So it's not just cosmetic. I think it's important. And anyway, we were not that lucky with Entostodon. Entostodon has a very high amount of incoherence among the different individual gene trees. We are leaving it at this stage like this. I mean, it's monophyletic, but there is a lot of conflict among the different individual gene trees. Last but not least, our friend Fiscomitrium piriforme, it's still polyphyletic, and at this point, we really wanted to do something about this. Okay, so two topics that we have pending. One, to resolve the status of this species that is very common and that apparently it has a lot of stuff going on there. And the second one, to deal with the polyploids. How are we going to deal with the polyploids? This is not trivial. As I said earlier, with Piper, you are going to be able to retrieve the different alleles for the same locus. That's great. So you are going to know for each of the locus which sequence comes from one of the parental subgenomes and which sequence comes from the other parental subgenome, the paternal and the maternal one. The difficult part here is to assign each combination of alleles to the right parental gene. Which ones come from the maternal side and which one comes from the paternal side. Computationally, this is very complex. Since the right combination has a higher likelihood, you can use a Bayesian approach that we call homologizer to just do the right match. But since this is, as I said, very computationally complicated, we had to reduce our data set from 660 to just 50 genes. Okay, so the last three that you're going to see now has a reduced data set with only 50 genes. And then, I don't know why I was so interested in this Fiscomitrium piriforme, but it's very, very common, right? So we wanted to sort this out somehow. The problem that we found is that people are not collecting this anymore, precisely because this is so common. If you try to find a recent collection in a herbarium of Fiscomitrium piriforme with spores that are viable for culture, you just don't find it. And at the same time, it was very frustrating because literally everybody could find it in the backyard, especially in North America. So we came up with this citizen science campaign in which we encourage people out there to learn a little bit about mosses and to pay attention to what they were looking. Thanks to the iNaturalist app that probably some of you are familiar with that here. We prepared this leaflet with just the basic information about how to identify Fiscomitrium piriforme, how to collect it at the right moment so that the spore would be viable, and ship it to us, to the University of Connecticut, where luckily they could be cultured and included in our analysis. Okay, so last iteration, probably you can't read anything there. I'm getting rid of everything that was already sorted out before. We have Entostodon and we have Fiscomitrium. You can't see it here because it's not included in this tree, but we got about several dozens of extra libraries from these very common species. And our conclusion is that there are actually 3 cryptic species under that name. So imagine that Capsella busa pastoris have 3 species and nobody had seen that before. So this is more or less what we are seeing here. 3 species that remain under the same umbrella. And very fast, I'm just going to mention a few of the allopolyploids that we found. One of them, Entostodon hungaricus, is an intergenetic polyploid. So the 2 ancestors are separated by more than 20 million years, and they hybridized and made a viable species of it, Entostodon hungaricus. We have other cases, such as these 2 Fiscomitria, that look very different, but they are descendants of the same cross. So it's the same parents, the same parental, well, the same ancestral species, but one of them has a particular maternal and parental ancestor, and the other ones are switched. And the result is that the morphology is very, very different. We have 2 more species, we do not need to talk about the details, that are also from hybrid origin. And last but not least, 2 samples that morphologically match Fiscomitrium piriforme. In this case, they are also allopolyploids. So not 3, potentially up to 5 different cryptic species under this very common name, which was actually the most that we sampled with more interest. Okay, I know this was a lot of information, so let's go back to the original questions. First, does the original traditional genetic treatment make sense? Well, no, right? We are moving towards a new treatment with about 6 genera, not the 16 that we originally had. And of course, it's very clear by now that all these syndromes of extremely reduced sporophytes, they are just a syndrome that has happened several times with no clear systematic meaning, but probably with a very high selective pressure that has been favored. Second question, and even more important, what is behind this loss of complexity? This was very perplexing at first if you think about that from a biological point of view. Well, we got some answers, right? Rapid diversifications, allopolyploidy hybridization, ongoing speciation events. All these features are the topics that fill the special issues of the new phytologies, the Journal of the Linnean Society. All these capabilities of fast evolving flowering plants are not exclusive from them. So bryophytes, or at least the Funariaceae, are enjoying of these capabilities that we find sexy and attractive in plant evolution. Of course, I would love to give you an answer of why is this happening? What are the selection pressures behind this extreme reduction? We don't know, but at least we can say that something that is structurally simple can be sophisticated. The next step, or something very interesting, would be to know what's the explanation of these changes. As I said, I don't have an answer, but some studies have been made comparing the transcriptome between Funaria and Fiscomitrella, and even in the generation that is almost identical in the gametophyte, up to 70% of the genes don't match. This probably means that despite the overall similarity, there is a lot going on that is not the same in Funaria as in Fiscomitrium. We are just missing that because we can only see what our eyes can see. Probably this is not only affecting the Funariaceae. we have evidence that other bryophytes can evolve very rapidly. So the lineages can be old, but most of the diversity that you see now, mosses and liverworts and homeworts are there, are relatively recent. And as we have seen, polyploidy, which is, again, one of these hot topics in plant evolution, is indeed present in bryophytes. We made an estimation, and if we took karyological diversity seriously, the increase in bryophyte species richness, diversity, would jump up to 35%, something very similar that you can find in angiosperms, or that people are talking about in angiosperms. Let's go back to this. So by now I want this to be like the take-home message. Bryophytes might be very simple. It doesn't mean that they are not complex in other ways. They are an alternative way of being a plant. And of course, we find our attention very easily drawn to things that are complex and that have a lot of moving parts that we see and enjoy. But we also know examples of things that apparently look very plain and very simple, but they also have a lot going on in the inside. And as a reminder for my 20-year-old self, bryophytes are actual plants, and they enjoy everything exciting that we find in botany. And despite being low plants, they are also able to reach high achievements. Thank you very much.
Thank you very much. Question. Hi, thanks very much for that. I'm wondering if there's any correlation with the recent or rapid radiation and biography, and whether that's important in mosses, because I know that they have really broad distributions generally, and maybe that means it's not important.
Yes, since they are sporic plants, if you check the books, right, the theory is that they can go from one continent to another. So long distance dispersal is going to, in theory, play a more important role than that. We haven't been able to make a correlation between that rapid radiation of the Funariaceae and any event that makes some sense, but there are a number of families of bryophytes that expanded also at the same time, the Miocene, I think it is. And there has been, I think it's just incidental evidence. I'm not very convinced by that, but some people made a correlation between some changes in the climate at that time and the expanse of these families. The other thing that I think it's important to keep in mind is that sometimes you do find bryophytes that are endemic of a particular area and that can have allopathic speciation despite being able to be dispersed by spores. So it's complicated. We don't have enough evidence to say anything in particular. Now, one of the possible future genera that you saw there, Fiscomitrelopsis, it's interesting because most of the species belong to areas with Mediterranean climate. Again, this is just incidental evidence. We haven't done anything there, but if I had to make like a wild hypothesis, maybe the origin of Mediterranean climate that had a very concrete moment with the when the North and South America got in touch, and then the water currents of the ocean changed, maybe there is some relation that when those type of climates appear, there was also like the origin of that particular genus. Yes.
So because there's such a diversity in this family, so we need to actually develop the rank to see it as a suborder or order.
Can you repeat the question, please?
Because there's such a diversity in this family, should we leave out the rank of this family to see it as a suborder or order?
The family itself has very clear synapomorphism. So I think that it's, of course, we can discuss whether the rank has a meaning itself, but the actual synapomorphy of the Funariaceae, regardless of these issues with the complexity of the peristome, is that the stomata, mosses have stomata, they are in the capsule, okay? Rather than having two cells, two kidney-shaped cells, the mitosis is never completed. So you have a stoma that looks like a donut. And as far as I know, this is the only family that has it, and this is a very strong synapomorphy. Traditionally, it has been a family. I think, I mean, no, there's no problem of keeping it a family. Yeah.
Thank you, Rafael. That was great. I have to admit that I've never thought about hybridization in terms of drug types, and I was just thinking at the end of your talk that in terms of the reproductive biology of the plants, what will prevent hybridization to occur between a really unrelated taxa? It seems that they will be really promiscuous, actually, right?
They are. We have to consider two different phenomena here. So there is a very good record on hybrids within the Funariaceae that are just hybrids, potentially sterile, and sometimes with, you know, a Funaria and a Physcometrium patens. So like the most complex and the most simple, and they will hybridize and they will give you a new generation. If they produce a sporophyte, it will be more or less intermediate. Some of these hybrids have been named and have been described, but the examples that I was showing there, those are well-established species that have an allopolyploid polygyny. I don't know when, you know, this hybridization will become successful in the long run and will produce something stable over the generations, but it's true that they are very promiscuous. So it's very easy to hybridize different ones. But most of the time, those hybrids will not, you know, be very stable, apparently.
I might have been misunderstanding one of your trees. Where you had all the monotypic genera, were they all most closely related to each other?
No.
One big group, it looked to me like it had all the monotypics. Yeah.
So I probably you mentioned. Sorry. No. This, right? So if the traditional genetic circumscription makes sense, we should see something like this, right? So every individual genus should be monophyletic, etc. I don't know, this was like one alternative. Maybe all these extreme reductions should be its own genus, and the reality looks not like this, but something like this. So we are moving towards perhaps 4 genera that have a good number of species, and then there are a few that have few species, but those are monophyletic. Okay, and most of the reductions, the extreme reductions, like the Physcometrella syndrome, those would be actually belonging to Entostodon or to Physcometrium. Did I make things clear, or?
I'm not really sure why there are so many monotypics.
Oh yes, okay, sorry. So I really don't know. It's like people were finding these reduced Funariaceae, and one of them would be in Australia, the other one would be in South Africa. And it's not just how the capsule would open. Some of them would have traits like, you know, like a swan-like neck pointing the capsule downwards, and these traits, and they would give a lot of importance to those particular traits and will erect a new genus just for that. When we entered the study of the Funariaceae, we thought that that didn't make a lot of sense. And at the end of the day, it seems that it didn't.
I know that Funn was trying to look for uses within plants, how they can be applied and why they got this setting. I'm wondering, does this family have any sort of broader ecological importance?
I didn't have time to mention the ecology, but these are they are usually present in very short-lived habitats. So, for example, Funaria hygrometrica, sometimes it's called a bonfire moss, because you will find in very nitrified areas after a bonfire. You will see that going on like there. Very frequently, you will see them in pools or in temporary water bodies that, as they get dry during a very short period of time of just a few weeks, the moss needs to establish from the spore and complete the life cycle in a very short period of time. And actually, that's perhaps the difference between Funaria and the other one, right? So the instructions for Funaria is like, okay, you have to release the spores when the sun rises at this time and the humidity is X, Y, or Z, and in this particular condition, then you release the spore. But for Physcomitrium patensis, you need to release the spore now, as soon as possible. Just do it very quickly, and that's it. So what all these ecosystems have in common, or most of them, is that they change very fast and they are temporary. Yeah.
And you really thought on what the last colonizers were planning plants regarding, or very possible anything close?
No idea, but if I had a time machine, that's what that would be the thing that I would like to. Yeah. So my paleobotany is terrible, so I don't know if I'm going to say anything that makes sense or not. But Cooksonia, which is a fossil that looks like a dichotomous stem branching all over, right? It's probably it evolved way later than the first land plant. But what I like about it is that it's isomorphic, or I think it's isomorphic, or it would bet that there is something very similar to a Cooksonia that is isomorphic. So the gametophyte and the sporophyte look very similar to each other. And that would explain why, when bryophytes have vascular tissues, they are in the gametophyte, and in vascular plants, they are in the sporophyte. During many years, it has been said that the hydron and the lepton, the vascular tissues of mosses, they are not homologous to the ones in vascular plants. I think they are because their genetic signature is there, but that would explain why you can keep it in the gametophyte and in the sporophyte. So this is just a wild guess, but I picture it as a branching series of stem-like cylinders, very basic.
For the new genus that you will describe, are there any good morphological characters that can be used for determining that genus?
Which one?
Like there are four, and then there is one like the sporation is very varied. Like, are there any other traits that can describe?
It's going to be difficult. Particularly, I mean, they shape so much that there is no trait there is no trait in the gametophyte that will lead you to the right genus. So you have to trust on the sporophyte, and it will be complicated because you have all these reduced forms that can appear in several genera. So if you use a typical dichotomous key, at this point, I don't know of any character that will make you reach the right genus just by using morphology.
How about the Antheridium? Are there any good characters?
I'm not sure. So what all the Antheridia have in this family, are you familiar with the Funariaceae? Do you know them?
Oh, no, but I'm just guessing, like maybe female part, male part.
I don't know. I mean, as far as I know, there are no useful characters there, but who knows? The Antheridia have some sterile structures that look like a club, and perhaps, but it's going to be tough. It's going to be tough.
Yeah, just wondering, you know anything about the sperm cell structure? Are there similar between the Funariaceae and Physcomitrium?
The spores?
The sperm cell.
The ultrastructure of the sperm cell has been very useful in bryophyte systematics. I don't know anything about the structure of this particular family, and I don't know if they will have any useful characters.
So right now, this new genus haven't been valid published based on the paper.
So at this point, well, so Funaria, you know, this was out of the. This is more or less what you will find, or what we are moving towards. The only one that has been formally addressed and the changes have made is Physcomitrium. This is what we currently call the Physcomitriolopsis clade. We are still working on this because we need a better sampling, but this is a candidate of becoming a genus. Just don't want to, you know, to spoil what is coming here. But it will likely be a genus. the bigger issue for us is Entostodon, because we are not sure yet if we can claim that it's monophyletic. But the only one that the nomenclature changes have been made is for Fiscometrium. And then the story is more complex, but I mean, there are a few other genera that don't appear here, but they don't pose any type of systematic problem. So 6 genera, that's something more reasonable that may be in the future, 6 or 7, something like that.
Any more questions? No. Well, thank you very much. That was great.